Our last speaker this afternoon is Rick Blumberg. Rick is a gastroenterologist, actually chief of gastroenterology at Brigham and Women's Hospital. He's a professor of medicine at Harvard Medical School. He has played very important roles in sort of the study of inflammatory bowel disease in the context of mucosal immunology, which I guess is his sort of disciplinary origin, and has done a lot of really interesting work that's already been alluded to recently in the symposium today on priority effects and windows of opportunity in the early development of mucosal immune system. So, Rick, thank you for being here.
Thanks, David, and thanks, Lonnie and James. It's a terrific meeting. And I also have an apology that I have an NIH study section tomorrow. I haven't done all my assignments, so I'm going to have to finish that up tonight and tomorrow. And also, I'm trained originally in infectious diseases in David's home, Mass General, and it was my recognition as an infectious disease fellow that all infections started at mucosal surfaces that inspired me to go in this direction. And so, as you'll see, that inspiration still resounds in the work that I do today. What I'm going to talk to you about is a clinical problem that I've been studying for about 20, 25 years, which is inflammatory bowel disease. And let me start here with this cartoon that summarizes the fundamental problem of these disorders. And we talk about inflammatory bowel disease, we're talking about two clinical phenotypes. We're talking about Crohn's disease, which affects the entire gastrointestinal tract. And in fact has extraintestinal manifestations. Some might think it's actually a systemic disease in which the GI tract is most prominently affected. And another clinical phenotype, which is ulcerative colitis, which is restricted to the colon and has autoimmune trappings, although not yet proven to be an autoimmune disease. I'll come back to that a little bit later at the very end when I wrap up. And it's really quite distressing for somebody like myself who's been studying this disease for so long that despite all of this molecular information you can see up here, we still define these diseases by these two basic clinical phenotypes. And the problem of both of these disorders is the relationship between the microbial milieu, which we've heard a lot about, the intestinal epithelial cell, which is a single layer of cell that separates us from the outside world, which is the size of, estimated to be the size of a tennis court, that sits on a basal lamina, and abnormalities of the immune system. And it's this abnormal tridirectional relationship which is the core of this problem. And each of these individual components, the microbial milieu, the epithelial cell, and the immune system, are each under the control of a variety of genetic factors, and each under the control of a variety of yet really to be defined environmental factors. And I actually would suggest to you that the thing that makes an environmental factor important in this disease, and probably all these diseases, but at least with respect to IBD, is that these environmental factors have to affect the microbial milieu, the epithelium, and the immune system in a very precise way in a particular genetic context. I'm not going to talk about the genetics because I'm sure Ramnik's going to give a wonderful presentation tomorrow. He's much more expert in that than myself. But what I'd like to talk to you about and focus my attention on is this abnormal relationship between the microbial milieu, the epithelium, and the immune system. And this is a very nice summary of the problem of the microbiota from a review that Jeff Gordon, it's actually the Cell Host and Micro paper, if you're interested in looking at this. And what Jeff does here is summarizes what the microbiota looks like if a patient comes to you with inflammatory bowel disease. So here's a patient that comes to you with inflammatory bowel disease. They've had this disease for years. You do pyrosequencing, you look at the microbiota, and you can find that in at least 1/3 of the patients, both ulcerative colitis and Crohn's disease, that there's demonstrable abnormalities. There's dysbiosis, if you wish, and the specific details don't matter in terms of the actual color coding, which is under here for the specific components. But there's abnormalities of the Bacteroidetes. There's a decreased, paradoxically, richness of this microbial milieu, which seems counterintuitive in a disease that's in part driven by microbes. There's altered composition of firmicutes, and there's a consistent, in all of the studies, bloom of proteobacteria. And you can find that some of the proteobacteria that bloom are pathogenic, that they adhere to the epithelium and promote inflammation. And there's a loss of organisms in some of these studies that are perhaps beneficial to the host. And the question that arises when this patient comes and presents with these abnormal microbial changes, or this dysbiosis, is this primary or is it secondary? It'd be wonderful it's primary because we could treat the disorder, but I refer you to a very important study from Brett Finley's lab, and there's a lot of other studies to support this notion, that one might surmise that actually most of this is actually probably secondary. And also, we have the data from Gerard, where he presented the review paper of the NF-kappa B disordered zebrafish, which has a dysbiosis due to abnormalities of NF-kappa B in the epithelium. And the same thing in this paper from Brett Finley, Luff and colleagues, is that a variety of different abnormalities of experimental abnormalities, whether it's genetic IL-10 deficiency, or it's environmental, that is dextrin and sodium sulfate, which affects the epithelial barrier, or pathogen, Citrobacter rodentia, which is an enteropathogenic E. coli homolog, all have a very similar effect on the microbiota and largely phenocopy what one sees in inflammatory bowel disease. And I see my colleagues over on the other side looking over there, so I'm going to start to I want to make you feel left out over there, so I'll start to move my pointer over there for all of you who are right-hand dominant in a few minutes, so bear with me. But the point is that a lot of this is probably secondary, and I think most of us in the community who study this think that most of what we're seeing is secondary. Now, is that unimportant? Of course it's not unimportant, because there's a lot of reason to believe that this dysbiotic state may be inflammatory in itself and perpetuate the disease. So, for example, in a mouse model from Lloyd Glincher's group, so-called truck model, where mice have abnormalities in Tibet and rag, there's a dysbiosis that's very similar that's developed, and this dysbiosis can be transferred to another animal and cause inflammation. The same thing by Flavel's group with the NALP6 inflammasome. So this is probably important, but it doesn't get us to what originates the disease. So in that regard, I'm going to open a page from the writings of Marty. I don't know if he's back yet, and remind us of what Marty said this morning, is that the diversity of the microbiome of a mammal and a human begins here at the earliest periods of life. And in fact, a child or a pup, a mouse pup, which we study in the lab, is born into the world and develops together with the microbial milieu. And they learn how to adapt together, and that there is an ontogeny of development of the microbiota, as we heard from many, but also at the same time, the microbiota is teaching the immune system, and the immune system is in turn teaching the microbiota in a way to form a certain community structure that's part of homeostasis. So what I'd like to then focus on then for the remainder of this is talk about that ontogeny and focus my comments, especially in these early periods of life, which I think may be very instructive for understanding the pathogenesis of these disorders. So I'm going to do that through the lens of a group of T cells that we've been studying in our laboratory for a very long time, which is the so-called natural killer T cell. And I'm going to show you a particular pathway that's very operative in inflammatory bowel disease and also important in terms of the ontogeny of the microbial milieu. And NKT cells are a very unique group of T cells that, in contrast to classical T cells that recognize peptides, NKT cells sense lipids. And those lipids are presented to the NKT cell by a non-classical class 1 molecule called CD1d. And these lipids may be derived from self, and there's a lot of autoreactivity in the system. And there's one very strong view, and supported by a lot of evidence, that NKT cells are running around the body actually sensing the lipid composition of cells and sensing the lipid composition of cells and deciding whether to respond or not respond. but also NKT cells respond to lipids derived from microbes. You don't have to turn it down. I'm afraid that I was falling asleep before. I know it wasn't the speakers. I know it was the light. So if you could keep it up, I'd appreciate it. Thank you. This is the only confocal I'm going to show. And so in the gut, there are two major classes of cells that express CD1d and restrict the NKT cells. And they're shown in green. And one group of cells are the professional antipresenting cells, which reside within the lamina propria. And they provide both self and microbial lipids to the NKT cells. And the other group of cells that provides a restriction for NKT cells are the epithelial cells, which separate us from the outside world. And epithelial cells have just as much of an importance in NKT cell biology in the intestines as do the hematopoietic cells. And in fact, in data that we have that's under review right now, we found that the CD1d on the epithelial cells is actually critical to maintaining homeostasis and actually orchestrating a pathway that is very important and just as important in our hands as hematopoietic cells for IL-10 production. But more to the point, NKT cells and CD1 have a very important role to play in inflammatory bowel disease. There's a lot of data from a mouse model in particular, which is the only NKT cell mediated model that we have, in that there's a hapten called Oxazolone that decorates both a host and probably lipid antigens, rather host and microbial lipid antigens, and stimulates an NKT cell response that is dependent upon CD1d and the T cell receptor of the NKT cell, because when you delete both of these, this response doesn't occur. And it involves both innate and adaptive cytokines. There's also data from a number of laboratories that NKT cells are important in human disease. Warren Strober's group has shown that in humans with ulcerative colitis, there's an enrichment in NKT cells that secrete a lot of type 2 cytokines. And in the recent, very large meta-analysis of genes that Skip was alluding to, where there's been identified approximately 163 genes and with containing about over 300 potential genetic elements, or 163 loci rather, that the NKT cells are interestingly enriched in the transcription profiles associated with the genetic factors that have been demonstrated to be associated with risk for the disease. There's a lot of reason to believe that NKT cells are a critical part of the biology of these disorders. And NKT cells, as I've already alluded to, have a very important role to play in responding to microbes by sensing microbial antigens. And this is a paper we published a number of years ago that a mucosal pathogen in the lung, Pseudomonas aeruginosa, the host depends critically on CD1 to respond to this, because in the absence of CD1, there's this dramatic overgrowth of Pseudomonas and severe pneumonia. Similarly, NKT cells are very important not only in managing the responses to pathogens, but NKT cells are very important to managing commensalism. So if you take a germ-free CD1d deficient animal and you monocolonize with E. coli, and then you compare that monocolonized CD1d knockout animal to a wild-type animal, you can see that at 9 hours, there's a 10 to the 9th greater number of E. coli colonized in the gut in a CD1d deficient animal than there is in a wild-type animal. You see the same thing with a variety of other organisms. So the message is that the CD1d NKT cell pathway is present, operative in the intestines, and very important in the management of pathogens and commensals. And in honor of Skip, I saw Skip was speaking, and to remind us of the importance of viruses in these disorders, that NKT cells are very important in sensing viruses. And this is a work we just published this past year, that in this case, the NKT cells are not responding to a pathogen-derived antigen, but the NKT cells are responding to a modified self-antigen. So what the NKT cells are actually sensing is abnormalities of the lipid milieu of the hepatocyte, in this particular case modeling hepatitis B virus, and responding actually to lysolipids and triggering a response that's orchestrating the entire T and B cell response to the virus. And in the absence of that, the T and B cell response to the virus doesn't occur. We would predict that the same thing is happening in a variety of other viral infections. So by responding to host and pathogen-derived lipids, NKT cells are able to initiate and control a variety of different inflammatory events. And actually, NKT cells are critical because they function at the earliest phases of the immune response, that is at the cusp between innate and adaptive immunity. So what I've shown you is that NKT cells and CD1 are very important in inflammatory bowel disease and in mucosal homeostasis and response to microbes. But the question I'd like to finish up with is, can the microbiota then in turn regulate CD1d NKT cell pathways, and what are the long-term consequences for the host? I'd like to walk you through some very interesting data that has, I think, important understanding, or has important implications for understanding the ontogeny responses, and asking or answering the question I alluded to is, when does, in fact, inflammatory bowel disease start, and to give you the message up front to suggest to you that it probably starts, or perhaps starts, very early in life, at the time of the earliest development of the immune system. And so the work, this work has been published, so I'll go through it rather quickly, but the sentinel observation was that mice that are germ-free have an accumulation of NKT cells in the colon, and very, very specifically in the colon, and to a lesser extent the small intestine. This occurred in the absence of any abnormalities of CD1 expression. And this process accumulated, this accumulation of NKT cells was pervasive and additive and increased throughout the life of the animal. So we asked the question whether whether in this context of germ-free state and this accumulation of NKT cells in the gut, whether there would be any abnormalities in a CD1d NKT cell mediated model. And the concept that I'd like to introduce to you is an old concept that I think is now breaking down. The old dogma, let's say 5 or 10 years ago, was no microbes, no inflammation in the gut. Well, that's just, I don't think that's really true. There's a lot of data that's been generated over the last several years, both in the innate immune system, as I'll show you in the adaptive immune system, but it's obviously a lot more complicated than that. And I think one of the very first studies was Leslie Metzitov's group, which showed that you could see a very exaggerated innate immune response in a germ-reduced animal with antibiotics. And more recently, Charlie McCabe's group has shown that germ-free mice, through the ability that are more susceptible to DSS colitis because of the absence of short-chain fatty acids, which they showed normally signal through GPCR43 and regulate the accumulation of neutrophils in the intestines. So there's already data to indicate that the walls, the fortress that was built up, that no microbes, no colitis, is really not correct, at least for the innate immune system. And for the adaptive immune system, we looked at this Oxazolone colitis model I've already alluded to, where you sensitize an animal and then you challenge them mucosally. And what was really shocking to us and surprising was that there was a dramatic mortality in the germ-free mice that have T cells and that have this accumulation of these NKT cells in particular in the colon. So this was an exaggerated T cell response in the colon in the absence of any microbes, showing that microbes were providing some kind of protection. It was therefore the first adaptive model that there was increased inflammation, and that microbes, again, were not bad, and they were not driving the inflammation, but through the lens of the NKT cell were actually protecting from the inflammation. And this is just showing that the mice had all the bells and whistles of cytokines that you'd expect to see in very, very severe pathology with, as Skip alluded to, trappings of human inflammatory bowel disease. And this inflammation was, in fact, CD1d restricted because you could block it by giving an antibody that was specific for CD1d. this is just all the supportive information, that in the mice, again, had an inflammation that was CD1d restricted, and therefore this was, as you would predict, a NKT cell-mediated process. But the really interesting point was the role of the microbes in this process. And the role of the microbes, and that was really what was most interesting, is that if you have a germ-free animal that has this accumulation of NKT cells, you can only normalize this phenotype by colonizing in the first 2 weeks of life. And there was a critical window of opportunity where the normalization of the immune system could only occur if the microbes are provided in the first 2 weeks of life, and not if the microbes are provided any later than that very critical window. And similarly, the susceptibility to inflammation also required introduction of the microbes in the first 2 weeks of life. And if this window was not, if the microbes are not provided during this window, this process was seemingly irreversible. And similarly, if the microbes were provided in this 2-week window, the protection was durable, because the environmental trigger that was given to stimulate this NKT cell activity to cause this inflammatory bowel disease-like activity was provided at much later points in life. And so this is interesting in the context of the hygiene hypothesis. And the hygiene hypothesis is worthy of note at this particular point in this discussion. And this hygiene hypothesis was originally raised by David Strachan in a paper in British Medical Journal in 1989. It didn't even actually use the word hygiene in the paper if you read it carefully. But it captured the idea that early life events were critical in defining later life susceptibility to a variety of allergic disorders. It was a birth cohort from 1958 followed for 23 years, and the children that were born in larger families or, in fact, were later in the birth order, that is, they were exposed to their older siblings, were protected from these disorders. It was later extended by Jean-Francois Bach to include autoimmune diseases in a very important paper. And there's an increasing amount of epidemiology. We heard a lot from Marty this morning, but increasing amount. I'm going to move over here to my colleagues to make sure that they know that I'm watching them and care about them over there, that I care about my left hand as much as my right. That there was epidemiologic information that early life antibiotics, we heard from Marty, predict the onset of asthma. In fact, antibiotics in the first week of life actually increase the risk for asthma in the next 5 to 10 years. And similar observations are made with inflammatory bowel disease. And I think a very important study which really inspired us was Erika von Mudius's group from Munich, Germany, who's an epidemiologist, showing the critical importance of early life exposures of German children on farms. And she's actually has specific microorganisms they've identified, microbes, not viruses, Skip. It may very well be. We should talk to her. That there are the critical bacteria that are at least epidemiologically linked to that protection. So in that light, it's very important to remember that inflammatory bowel disease is a disease that, like all these diseases, is increasing rapidly throughout the world. This is a terrific epidemiologic study I would refer you to if you're interested in this area. It's a retrospective review, but it shows virtually every study that's looked at the epidemiology has shown the increase. I think most important, which is this, is the title of this paper suggests that these diseases are now global diseases. And the paper pointed out the emergence of IBD in areas of the world that traditionally, if you read old textbooks of medicine, have been thought to be resistant, if you wish, or not sites of IBD in areas in Asia, Africa, South America, and the Middle East. And I go around the world, I talk to my colleagues all over the world, and no matter where I go, people talk about these diseases popping up. And it's very interesting that the group of people that may be most susceptible to these diseases in the increasing incidence are the children. And this is a very interesting paper that's showing the kids in the first 5 to 10 years of life are the ones that are showing the greatest increase in incidence. And there's this group of disorders that my pediatrics colleagues call early onset IBD. Some of it, which occurs in the first few years of life, is clearly Mendelian in this environmental context. But there seems to be increasing incidence of sporadic inflammatory bowel disease that's increasing as well. And another group of disorders that, and Marty alluded to this morning, that are also increasing are the atopic disorders such as asthma, eczema, and hay fever. And asthma, in particular, has been linked to the activity of NKT cells by Dale Umetsu when he was at Stanford. And in this paper, we looked at asthmatic models in the lung, and we phenocopied exactly what we saw in the gut. So in the lung, there was an increase in NKT cells infiltrating the lung, and it can only be normalized if the microbes were provided in those first 2 weeks of life. Those animals were also susceptible to allergic asthma using the ovalbumin hypersensitivity model, and this can only be normalized in the first 2 weeks of life. And so we began to look at mechanism, and I just want to quickly finish up in the next 5 minutes just talking about some mechanism. And we looked at a chemokine ligand called CXCL16, which had previously been shown to be a ligand for NKT cells, and I'm going to move over here now to CXCR6. And this has both a transmembrane and a soluble form, and it regulates NKT cell chemotaxis, and has previously been shown, although they didn't realize the relationship with NKT cells in these papers, to be a surrogate marker for the activity of a human inflammatory bowel disease. So it's quite interesting for us to look at this, and this is a really very interesting experiment because it shows that a mouse that's born to an SPF, a specific pathogen-free mom, or a mouse that's born to a germ-free mom, are born exactly with the same levels of CXCL16 in the gut. And yet, the germ-free mice then increase their CXCL16, and that means implying that the microbes are repressing the CXCL16 and the infiltration of the NKT cells. And you can block this entire process by blocking CXCL16. So if you give anti-CXCL16 antibodies to the mice in the first 2 days of their life, as Torsten Olzak did, you can block this entire infiltration. Thirdly, the CXCL16 is increased in the mice that are germ-free, can only be normalized by providing a microbial signal in the first 2 weeks of life. The signal is coming, at least in part, from the colonic mucosa and immunolocalizes to that interface between the outside world, the epithelium. And you see the same thing in the lung, that it immunolocalizes. There's at least some of the signals coming from the lung and immunolocalizes to the epithelium. And CXCL16 had previously been shown to be expressed by the epithelium. So this was all consistent. And the last point is that what the microbes seem to be doing is regulating through a pathway we don't understand yet the epigenetics of CXCL16 and its expression. And so, collaborating with colleagues in Kiel, Germany, who are expert in this area, we identified 5 potential sites on the CXCL16 gene that were amenable to methylation. And what they observed counterintuitively was that there was hypermethylation of that locus in the germ-free mice. And we always think about the methyl signature as turning off genes. But in this case, it would appear that it was turning on the gene. And again, this hypermethylation signal in the lung and the colon were only normalized if the microbes were given in those first 2 weeks of life. So there's a critical window for epigenetic modification of the CXCL16 gene. And it turned out, we looked at a signature that had recently been described in the last several years as an activating methyl signature. It's called hydroxymethylcytosine, 5-hydroxymethylcytosine. And the CXCL16 gene seems to be marked by this signature, and again, was only normalized when the microbes were provided in the first 2 weeks of life. And we could then start knowing this, we could start to play with this and the environmental signature, or the environmental cues. And so we could give very high dose folinic acid to a specific pathogen-free animal and force the methylation of CXCL16, force the expression of CXCL16, and force the infiltration of the colon with NKT cells. And it's interesting because folic acid in the last trimester has been suggested, controversial, to be a risk factor for asthma in certain individuals. There's studies for and against it. So again, are there other environmental factors that are cooperating with the microbes to do this? And similarly, we could give 5-azacitidine to the germ-free rodents and suppress this. and inhibit the CXCL16 and inhibit the invariant NKT cell infiltration. And so I'm going to take another page from Marty's work, and he showed this slide this morning, is all these critical environmental factors that are happening or occurring in the earliest parts of life in mammals. I also want to refer you to some of David's work, David Relman's work. There's a whole body of work in this area just showing, and Marty talked a lot about this morning, about the critical role that antibiotics play in what he and Stanley Falco called lost friends. And I've already told you that antibiotics as well as C-section are risk factors for these diseases. And so I just want to finish up with one last experiment, which is that we could show that antibiotics could also phenocopy this in the first 2 weeks of life. In this particular experiment, what was done was a specific pathogen-free mouse was given a single antibiotic. A pregnant-specific pathogen-free mouse was given sulfatrim. And the at the very last days of pregnancy. And the sulfatrim was continued in the mother, so it was transferred through the breast milk for the next 2 weeks and then stopped. And then we examined the pups at 6 weeks of life. And we could see that in those pups that received sulfatrim in the first 2 weeks of life from their mothers, that there was an accumulation of NKT cells in their colon. There was an increase in CXCL16, which did not depend upon the presence of CD1d or the T cell, the NKT cell, because in the knockout mice, the CXCL16 was elevated, meaning this was a cell-intrinsic effect, presumably directly on the epithelium, and that the mice were highly susceptible to this NKT cell-mediated colitis. So what this shows is that an environmental event that degrades or diminishes or abrogates the important microbial cue in the first 2 weeks of life allows the animal to become susceptible to an environmental trigger later in life that leads to the activity of this disease. And so this is, to sum this up, and that this is a model of what we think might be going on, is that there appears to be a very important relationship of the microbes. NKT cells, I should say, come from the thymus. They're born in the thymus, and they migrate to the peripheral tissues. And it would appear that the gut microbiota are critical for repressing one of the important physiologic environmental cues to their infiltration of these tissues, at least in part through a mechanism that needs to be defined, the epigenetic modification of this particular locus. And in the absence of this, there's a inappropriate expression and enhancement of CXCL16. And that the microbes further this would therefore lead to a regulation of the infiltration of the tissue. But we also think there's a second event. And that second event we think is actually CD1d restricted and antigen dependent. This part, as I've already shown you from the antibiotic experiment, has no relationship to CD1d. But we think there's another part here, because recent work from Dennis, who has been a very close collaborator on this project, and I'll say that at the very end, Dennis, in his laboratory, has just recently identified a glycosphingolipid from Bacteroidetes fragilis that inhibits this entire pathway. It inhibits, it can actually phenocopy many of the findings I just showed you, such as the early life sensitivity to regulation and susceptibility, early life regulation that leads to prevention from susceptibility to later life triggers that activate NKT cells and inflammation. And Bacteroidetes fragilis that has been engineered by Dennis to lack this property are unable to provide this antigen-specific recognition, suggesting that there are commensal microbes that provide CD1d-restricted lipids and participate in this regulation of NKT cell infiltration into the colon and regulation, lifelong regulation of this pathway, or apparent lifelong regulation of this pathway. And so with that, let me just finish up and mention the most important slide, which is the people who did the work. This is Torsten Olzak, who's now setting up his own laboratory in Munich. Important help from Becky Barron and colleagues from the asthma. And this was really a very close collaboration between Dean and Torsten and his lab. We continue to work on this together because I suspect that, and our hypothesis is there are a variety of other commensal microbes that have sphingolipids that are CD1 restricted and are probably have the same basic effects in these early life events. And there's going to be a lot to discover here. with very, very important, hopeful translational implications. Thank you very much.